We're going to walk through the RNA-seq analysis pipeline step by step, just like solving a biological mystery. We start with raw sequencing data, and our goal is to transform it into meaningful biological insight. Along the way, we'll clean the data, map it to the genome, count gene expression, normalize it, and finally discover which genes are truly changing. Think of this pipeline as turning raw signals into a clear biological story. Before we trust any data, we must check its quality. In this step, we use tools like FastQC and MultiQC to examine read quality, GC content, and adapter contamination. Good data should have high base accuracy, typically Q30 or above, and a smooth GC distribution. If we see problems, like low quality or adapter contamination, we fix them by trimming reads. Because if the input is bad, everything downstream will be unreliable. Once the data is clean, we align the reads to a reference genome. This means finding where each sequencing read originally came from in the genome. Tools like HTSeq or STAR help us do this efficiently. The output is stored in SAM or BEM files, which contain detailed information like read position and alignment quality. This step connects raw reads to real biological locations. Now we convert aligned reads into numbers. We count how many reads map to each gene using tools like featureCounts or HTSeq. This transforms our data from alignment files into a gene expression matrix, where each number represents how active a gene is. This is the first step where biology becomes measurable. But raw counts are not directly comparable. Different samples may have different sequencing depths, gene lengths, or composition bias. Normalization corrects these issues so that differences reflect real biology, not technical artifacts. Methods like TPM, DSeq2, or TMM help make samples comparable. Without normalization, comparisons would be misleading. Now we ask the key question: which genes actually change between conditions? Using tools like DSeq2 or EGGR, we identify genes that are significantly upregulated or downregulated. This step combines statistics and biology to distinguish real signals from noise. The result is a list of genes that may explain the differences between conditions. Finally, we turn results into meaning. We analyze pathways using KEGG or gene ontology and visualize results with heat maps, PCA, and volcano plots. This helps us move from a list of genes to understanding biological processes, mechanisms, and potential discoveries. In the end, RNA-seq is not just about data, it's about telling a biological story. From raw reads to biological insight, RNA-seq allows us to uncover the hidden mechanisms inside cells, one step at a time.